everyone. In lecture 1, we start talking about biological background related to bioinformatics. And from today, we will talk about the algorithms that we need in bioinformatics and also data analysis that you will see later. But today, let's deal with pairwise sequence alignment. And in this picture, actually, you see the multiple version that we will talk about it later. And we will mention that why we need such sequence alignment and why they are useful for bioinformatics. But before dealing with that, let me introduce to you some important terms related to sequence alignment. And generally, when we talk about sequence alignment, we are looking for similarities between two genes or, for example, two proteins, and we will talk about them in details why this is necessary. So what about these terms like homolog genes? Let me introduce you these terms in this picture. We can understand about them better. When gene duplication happens, which is the reason that we are alive, because, for example, we don't say a table is alive because it doesn't have something like gene duplication. It cannot duplicate its gene. That's why we are alive, so that we can transfer our information from one generation to another, and through it we have evolution. So the history of evolution, which happened for thousands of years, for millions of years, that actually made what we are now, and that made the variety that we have in our world. So this evolution happened so that we have different organisms, like we have human, we have fish, we have mouse, but they have some similarities because they inherited some information from the same source, and that's why we know that there are some similarities between the genes of different organisms. So gene duplication happens, and through it we may have mutation in the gene, and it can changes many things in that gene. And through evolution, if it happens a lot, then we have totally different species after a while. It can happen, but to have a species is more complicated. Sometimes a huge part of a chromosome could be mutated, or the whole chromosome could be missing in some species, or maybe added. So many things can happen, and actually I will talk about them in details later. But the thing is that because of those mutations, then we will have different kind of genes. And like, for example, alpha chain gene and beta chain gene, usually call it alpha globin gene or beta globin gene in human beings, and they do pretty much the same function, but there are, of course, some differences, but we can see some big similarities. It shows that they are coming from the same father, if I would say. So alpha chain gene and beta chain gene, which creates alpha chain protein and beta chain protein at the end. So now assume that we have alpha and beta, which we call homolog, because they have pretty much the same function. The gene sequences are somehow similar, but there are many differences inside them as well. In order to call them homolog, again, we will talk about it in detail in which measure we can use to say, okay, these two genes are homolog. And let's go through more details. Like when we look at different organisms, then we will see that, for example, in a mouse, we have something similar to alpha chain, and we call it mouse alpha gene, and human alpha globin gene, for example. And we see there are some similarities between the function of these two, and that's why we call them ortholog. So ortholog is a kind of homolog gene. So we may call them homologous or homolog genes. These are the terms that they use a lot. So in this case, when you see very similar genes in different species or different organisms, then in that case, we call them ortholog genes. And sometimes in the same organism, for example, in a mouse, you can see alpha and beta. And these two are very similar and they have pretty much the same function. Then we call them paralogs. For example, in human beings, we have alpha globin gene and beta globin gene, and both of them are responsible to separately synthesize alpha globin protein and beta globin protein. And these subunits comes together to have a hemoglobin in our body. So at the end, we will have hemoglobin because of these two genes. And it happens through evolution that these two came together to make it happen. Maybe in previous species, we didn't have such a case. We have only one gene. We call it, for example, homoglobin gene, and that one was responsible to do that. But through history of evolution, many things happen, and we have these variety, as you can see here. So now for you, it's important to know what is homolog, what is paralog, and what is ortholog. So by looking at this picture, I think you can always remind yourself about the similarities and differences, which is very useful. And there are some databases that you can use for sequence alignment and also about comparing genes together. And they also mention about these homologous genes as well. You can find in these two databases. We will talk about them later. And we have a term named conserved region. Through history of evolution, as I said, many changes happen, maybe because of mutation. And some part of the gene, if we look at it, or even DNA, then you will see it reserved the same. There were no changes. For example, if you check it with a mouse and also check it with you see exactly they are the same sequence in some part of it. And that's why we call it conserved region, because through evolution, it was no change in them. Like, for example, many of us have eyes. So the function of eyes are pretty much the same, right? So maybe there are some differences, but some part of it that we are able to see something. So this is the function of eye. And some part of DNA, maybe it's exactly the same for us, and it was conserved. No changes through history of evolution, and it remained the same. Right, so here I listed some important notes for you. Like, for example, more than 400 genomes have been sequenced so far. I told you it's very hard to find the genome of an organism. It's not an easy task. There are big projects for that. So far, more than 400 we have, which is a big deal. But still, we can have much more than that, and they are working on it. And the lengths of genes are not necessarily the same in sequence alignment. So when we compare two different gene sequence or protein sequence, not necessarily the length of them are the same. And how we can compensate that using gaps and using other methods, we will talk about. We have terms like pairwise or multiple sequence alignment. When you compare only two sequences together, then in that case we call them pairwise, which we will cover through this week and next week. But sometimes we are dealing with multiple sequence alignment, which is very useful as well. The picture I provided for you at the beginning was related to that. And we will talk about it later for sure. And we have terms global and local sequence alignment. When we talk about global one, it means that we are comparing two sequences together with all of their components. Like, for example, you are comparing all the nucleotides together, or maybe you are comparing all amino acids together. But sometimes we are interested only in a specific part of gene or a specific part of protein, which we will call domain, and I will show you. And we are trying to compare them together. Then in that case, we call them local sequence alignment. today we will talk about global version more, and actually we will continue it on Wednesday about global sequence alignment. But the term I mentioned about domain was a part of protein which is responsible for a specific function, because the protein maybe has multiple tasks. It can do many functions. That's why we can separate them sometimes into different partitions and call them domain. And you can find such domain in domain databases, which we will introduce to you later as well. So for the time being, during these 2 weeks, I'm introducing to you many terms. It would be confusing for you. That's what we are going to do next. But they are just introduction. Then later, when we are using them, and you will see your weekly assignment, then you will see why we need them, why we need to know about these things. And there's no need to memorize all of the terms. Automatically, it will happen. Whenever I ask you a question about, for example, a domain, then automatically you should remind yourself what was the domain, and we already introduced to you during the 2 first weeks. And the important question that arises when we talk about sequence alignment. Why do we need them? So the first reason is that to find evolutionary relationships between organisms. So I already showed you a picture related to homolog genes, and you know that it's important in order to organize them, in order to categorize them in different groups, so that it will make our job much easier. But for what? For example, protein function prediction is one of them. You can predict what a protein does, which is very important. We don't know still, for example, all the genes in our body is exactly responsible for which function. We have already discovered many functions for each protein, but still we cannot for sure say we know all the function of this gene or protein. So this is an important consequence of sequence alignment. And shotgun assembly, what do we mean by that? Because as soon as you understand about the function of each domain in the protein, then assume that we can assemble them together and we create a new function for our protein. And this is possible. And it's not as easy as it sounds. I'm oversimplifying things like just assembling different proteins together. In molecular biology, it doesn't happen easily like that. In computer science, we can easily just cut wherever we want and just add sequences together. But we should be careful in molecular biology that it's possible or not. And we should know about the structure of proteins, that even this is physically possible or not. And of course, we will talk about them in details. Finding motifs is also one of them, which is very close to the prediction of gene and proteins. And treatment prediction, which is the most important, I think, the application of sequence alignment. Because if you understand which genes are homolog together, because we can still claim that we know all the homolog genes, or we discovered everything. And that's why if you find such similarities and see their 2 homolog genes together, you discover a new one. And it will be a big step for treatment prediction. Like, for example, for hemoglobin, that they already done many research. They know, for example, gamma globin, which is present in an fetal human. Then we can upregulate the gene expression of this gamma globin in patients of thalassemia patients. So in that case, we can compensate the lack of, for example, beta globin, which should be present in adult human, but it's not for thalassemic patients. So these kind of things are very important in order to find novel treatment for different disorders or different disease. So let's go through details of pairwise sequence alignment and how it happens. First, we start with very simple ones, and not officially I even introduce you an algorithm, but step by step we are tackling it. And from next week, I will also show you some important algorithms that they use. But this session is very important in order to understand what is a pairwise sequence alignment. Okay, consider these two sequences. So there are nucleotides. It's like we are comparing genes together, not proteins. So you see the length of the first sequence is longer than the second one. And we aligned, for example, the third component or third nucleotide of this gene to the first one of the second sequence. Or you may ask why we didn't align this one, this T, with this one. So these are the questions actually which arise later, and it will get even complicated that which one should be used. In this picture, it seems easy that yes, the first one we just align with this one, but it's not always the case. Maybe I made a mistake if I align this one with the other one. As I showed you, there are some parts of the DNA we call junk DNA, and if you align such things together, it will be meaningless. And there's another problem that we have some spaces here before and after the sequence. And what happens to G and C? Should we just throw them away? Or can we do something before and after this sequence? And that's why we introduce gaps. You see? We can show them by dashes like this. So we align G with a dash, C with a dash, and they are meaningful later. We will talk about them, why we do that, even they are meaningful. And it's not wise to align one dash to another gap to another one. It's meaningless because later when we introduce you scoring function, you detect similarity by mistake. So it's not a good idea to align two gaps together. So it should be a letter to a gap like this. So you see the red color represents the one that we align with the other nucleotide from the other sequence. All right, so let's see how we can compare the sequence alignments together. Like we have exactly the same sequences, and which one would be the best alignment? Like here you can see the sequence is exactly the same with this one and this one. The second sequence also the same. But you can change the position of them. You cannot change the position of letters. The order should always remain the same. But you can shift it forward and backward. And now, for example, we align the first letter of the sequence with the second one. In the next one, we align the second letter of the first sequence with the second sequence. So which one is better? That's the question we should ask. And that's why we introduce scoring function to decide which one is the best alignment. Like this is an example to show how we can do it. But you know, there are different algorithms and no one can claim my algorithm is the best because they are still trying to find the best sequence alignment. And sometimes, you know, in some cases, one algorithm works very good, but in other cases, don't. That's why we should learn about different algorithms to know which one to use for which type of sequences. And for example, you see AGC is the same sequence as AGC here. These are the gaps that we are introducing manually ourselves. It's not like in our gene we have a blank, for example, space like this. We have AGC exactly in the sequence in our gene. Yes, of course, it can be a deletion mutation, which I will talk about later. But even if deletion happens, AGC will stuck together. It's not like we have empty spaces between them. These are the ways we show in computer science in order to compare the sequences together in bioinformatics. And these are the things that we are doing. In nature, we don't have dashes, for example. Or we don't have, of course, letters. There are chemicals in our body. Okay, so we have such sequence alignment that we applied such gaps between them one by one. And here, these two gaps are together. And we align this one with this, and this one with this, you see? And C is not the same as T, so I didn't show it with red color. And as you can see, we have two red colors here because exactly they are the same nucleotide, and here the same story. So if I want to compare together as the number of matches, then in that case I say, okay, +2 this one, +2 this one. So these two alignments are exactly the same. But you know, in nature, it rarely happens that we have such a mutation that one by one we have deleted one nucleotide like this, and we got AGC together. It rarely happens, and it's possible that we have deletion like this. Suddenly we lose some information about few nucleotides together, comparing to one by one. So in that case, if I want to show that which one is a better sequence alignment, it's better to choose this one over this one. It's not 100% sure, maybe we made a mistake, but this is more probable that we have such a case. Okay, so in that case, when I want to define my scoring function, it's better to give some negative points to this one more than this. For example, if I say I want to analyze this scoring function, I can count the number of gaps, which is 4. Here is also 4. If I say -4, then 2 minus 4 would be -2. And again, 2 minus 4 would be -2. Exactly the result would be the same as the scoring function. I want to penalize this one more, penalize this one more. So how can I do that? Maybe I give -2 to each one of separate gaps like this. So if I want to give penalty for this sequence alignment, I will consider -2, -2, -2, -2. Together, -8. So 2 minus 8 will be -6 as the total scoring function of this sequence alignment. Here, if I penalize this one with -1, then it will be -4 altogether. So 2 minus 4 would be -2. So you see, -2 is bigger than -6. And because of that, I could say this one is the better sequence alignment comparing to this one. So you see, it's in our hand. But how can we define the best scoring function? That's the question. And that's why we should introduce you different sequence alignments, different algorithms related to them, and you will see which one could be the best in a specific cases. And now we just casually introduce you what we are doing and how we can define it. For example, in the next weekly assignment, I will give you one example related to this so that you can see how we can apply such sequence alignment and a scoring function easily. But later, when we have very big length of sequences, then it's not easy to apply it manually. And that's why we need computer programs to help us with that. We need simulation. And we will talk about them later. So we already talked about global sequence alignment, that we have two strings, like sequences. And here you can see the official definition of global sequence alignment, which we talked about. We are comparing the whole sequence with another one. And why we call it pairwise? Because there are two strings. If there are more, we call them multiple sequence alignment. So we talked about also sources of variation through gene duplication, and why we have variety of organisms in the world, in our universe. So one of them is nucleotide substitution. means that if you have a sequence, then we have a mutation, and instead of A, we have C. Or for example, instead of C, we have G. So whenever such things happens, we call them nucleotide substitution. And when we have insertions or deletions in DNA, it means that some letters in our sequence are removed by mutation, or some letters are added. I'm using letters just as a terms of computer science, you know, guys, by now. So, you know, in reality, it means that we don't have some nucleotides missing or added by mistake. Or we have duplication of single gene or chromosome. So a single gene could be duplicated, and it will be a big problem, and it can change the function of the protein completely. Or even if we are talking about chromosome, that's a big change. That's a huge change if such a thing happens. And maybe there are just some part of a gene and part of chromosome. Again, it could lead to big differences, big variation. And if this is the whole genome, then we have a totally new species. Like suddenly we have human instead of a monkey, you know? So, but you know, these things doesn't happen overnight. It happens over so many years in history of evolution. You should know about the sources of variation, and even a single nucleotide substitution. When we understand this can lead to a disorder, so we take everything very seriously here. Can we have, for example, a mutation could be useful for us? For example, what we see in science fiction movies. Yeah, in theory, yes, they are possible. But how easy is that to substitute it ourselves in our genome? Yeah, that's not an easy task. They are, of course, working on it. There were many successful treatments, as they call them. But you know, many things maybe happens in such experiences that are confidential, and we don't know, for example, what they are experienced on their subject. But anyway, as much as science goes, and it is our concern, we introduce you usually the disorders and such things. We don't go through the science fiction things, you know. But you should know about the sources of variation. And there are some common assumptions in our sequence alignment, which are not totally true or even completely wrong in the nature. So I listed some of them for you here, that we have such common assumptions, and we neglect actually some of the things which are in nature. But we are doing these things because we are making such sequence alignment algorithms. It makes our job much easier, and it's helpful too. It's not just like we are lazy to simulate the nature, but we are doing our best to make these algorithms as closer to nature as possible. You know, in mathematics, we do such things often. It is true that mathematics is based on logic, based on facts, based on theorems. But time to time in mathematics, we say, for example, we neglect this part, or we estimate this part, especially in statistics. You see it a lot, that we have errors, and we say, for example, 95%, I'm sure this formula works. So we have such things also in sequence alignment. We can even find error, and we can compare them to the nature to understand how much we were close to predicting what's happening in nature and how we are far from it. And these are our challenges as bioinformaticians to deal with that. Okay, guys, so on Wednesday, I will talk about in details related to gene mutation and also scoring metrics, which is based on the scoring function that we talked about today. So we will deal with that on Wednesday briefly. And from next week, we will start our weekly assignments and I will introduce you different algorithms related to sequence alignment. So please, if you have any questions, let me know. And I hope it was a useful session for you.